Hi guys, this is chapter 22. We're going to talk about the reproductive system. The first topic in the outline is basically just a review of our gametes. So let's just take a look at this flow chart. Gametes are haploid sex cells, so they're going to be involved in producing a genetically unique individual, our offspring. And in both males and females, we are born with stem cells. So there are stem cells found in the gonads. Remember, the word gonad is for the organ that produces these gametes. And the stem cells for males are found in the testes, right? These are in the testes. And the female stem cells are in the ovary. Those are the gonads. So these guys are 2N. They are diploid cells. 2N. Okay, so the word diploid. So let's just review real quick. What does N stand for? N stands for the number of different chromosomes that we have. Different organisms have a different N. Humans have 23 as our N. So our chromosomes, we have chromosome number 1 all the way to chromosome number 23. So we have 23 different chromosomes. A quick word on the different kinds of chromosomes we have. 1 through 22 are considered autosomes, because they do not determine sex. But the 23rd chromosome is considered a sex chromosome because it does determine the sex of the individual. So the sex chromosomes are either X or Y. Okay, so when the stem cells undergo gametogenesis, so the process of making gametes is gametogenesis, and this, remember, is meiosis. The male gamete are spermatozoa, so the N is 23, they're haploid. The word haploid is that they only have one set of chromosomes. The female gamete is an ovum, all right, also haploid. And when there's fusion, when there's fertilization occurring and a sperm fertilizes the egg, then the resulting cell is a zygote. The resulting cell is then 2N. So a 2N cells, the word for 2N is diploid, again. And you have a whole other set of chromosomes. So you have 1 through 23 inherited from the father, from the sperm, and 1 through 23 inherited from the egg. All right, so let's take a look at, so the other terms in your outline are that an embryo is what you call the developing organism from the first 2 months, and then after that, after the 2-month mark, then you call the development of a fetus, developing person, a fetus. Okay, so this is just a look at our chromosomes here. The X chromosome is much larger than the Y chromosome. The X chromosome also contains other really important genes, so it has essential genes on it. So both men and women, or males and females, require at least one X chromosome. The Y chromosome is especially important in men because it has genes that are responsible for developing into the male phenotype. And we'll look at that in just a second. So just remember that the genetic combination for female is XX, and the male combination is XY. All right, so we're going to take a look at this picture here. So this is what, I'm going to blow it up a little bit for you guys. In the first couple weeks of development, there are both Wolffian ducts, Müllerian ducts, and primitive gonads. So these are undifferentiated structures. The Wolffian ducts are the precursor to the male reproductive tract. The Müllerian ducts are precursors to the female reproductive tract. So this is the male. So you can think about males as wolves, or werewolves are mostly depicted as being male. And then Müllerian is going to be your female precursor. So when we have, so this is the importance of the Y chromosome. The Y chromosome contains a gene that is called the SRY gene, which is called the sex-determining region on the Y chromosome. That's how it got the XRY name. And it codes for a protein called testis-determining factor. So in the presence of, so for the male development, the male is XY. The Y has the SRY gene, and that's going to create a protein called the testis determining factor. Okay, and that's going to make the gonad, right? So where we have a primitive gonad that's undifferentiated, it's going to develop into testes because of this testis determining factor. So we have testes developing. Now that we have testes developing, the cells are going to be prompted to make testosterone and another hormone called Müllerian inhibiting substance. Okay, so that, oops, sorry about that. So the testis is going to make testosterone, and it's going to make Müllerian inhibiting substance. So because of these two hormones, the testosterone is going to help promote the development of the Wolffian ducts. And the Wolffian ducts, let's just move down to 7 to 8 weeks of the embryo. Oops. The Wolffian ducts will then become the male reproductive tract. The MIS, the Müllerian inhibiting substance, is going to act to degenerate the Müllerian duct. So that just basically goes away. It atrophies and then it completely disappears. And that's how you get the male phenotype. If you look at the female pathway, okay, the female develops, they're going to inherit two X's. So there is no SRY gene, right, because we do not have a Y chromosome. In the absence of this SRY gene, in the absence of testis determining factor, the default is to become an ovary. So the lack of the testis determining factor means that these cells, these gonads, are going to become an ovary. Now, also, because we're not producing testosterone and we're not producing the Müllerian inhibiting substance, then we're going to have the development of the Müllerian ducts, which I don't have right here, but it's going to develop, the Müllerian ducts are going to develop into the female tract, okay? And then because there's no testosterone to prompt the Wolffian ducts to stay around, the Wolffian ducts are going to also atrophy and go away, okay? So it's kind of a funny thing that it's basically in the absence of signal, the undifferentiated organs will become female. But in the presence of the testosterone, in the presence of the MIS, right, you're going to have the male phenotype. So that's basically how we have sex determination and sex differentiation. It's through genetics and it's also through hormone control. And we also need to think about that the hormone receptors have to be functional, right? So they have to be able to detect testosterone and they have to be able to detect the Müllerian inhibiting syndrome. So if there's a problem in this early range of development, it is possible that a person is XY genotypically male, but if there's a problem in testosterone production or if there's a problem in testosterone receptors, then you could actually feminize and you can have an XY female. So sex determination and sex differentiation is a little bit more complicated than what we're going over, but that's one factor that can be, that is out there, that there are XY female, phenotypically female people out there. Okay, so this is just a flowchart of what we talked about so far, right? So what I want to bring up here is that we have the sperm. So here's our male development, and this is our female development, right? This is our male, this is our female. Notice that the eggs are all both X, right? So a female, a woman, is genotypically XX. So she can only donate when she creates an egg. The only sex chromosome that she can give to the egg is an X egg, right? So these guys split up. There's an X in each egg. Men, however, right, males have an XY genotype, and so when they produce sperm through spermatogenesis, they can have an X sperm or they can produce a Y sperm. So it's really the male that determines, the sperm that determines the sex of the baby, whether the X or Y chromosome fertilizes the egg. So if the Y chromosome is the one that fertilizes the egg, the zygote, then we have the presence of the SRY gene. We have that testis determining factor, right? Testis determining factor being produced. And we're going to get testes being made because we have testes made now, we have the presence of testosterone and the presence of another hormone, this MIS, Mullerian inhibiting substance. The testosterone is going to kick off the production of the Wolffian ducts, which eventually become the male reproductive structures. The inhibiting substance here inhibits the Mullerian ducts and promotes this regression and atrophy, and it goes away. Okay. In the female column, we have an X egg and an X sperm, making an XX zygote. We do not have the SRY gene. So the default, those undifferentiated gonads, in the absence of a signal, they're going to become ovaries. And the ovaries are not going to produce testosterone, right? So in the absence of testosterone, the Wolffian ducts don't develop, they're going to regress. In the absence of MIS, if you're not inhibiting the Mullerian ducts will go ahead and mature, and they're going to form those female reproductive structures. All right, so let's move on to the next topic. This is just the patterns of reproductive activity over a lifetime. So you already probably know this, that adolescence is considered about before 10 to 14, so as you're a kid, right? The reproductive organs are not fully functional yet. They're not able to have, the gametes are not there, they're not mature, they're not going to be able to be fertilized, so these adolescents are not able to have children. Their reproductive system is not mature. At puberty, this is where that shifts, right? So puberty is anywhere from 10 to 14, the onset of puberty. And you're going to have a dramatic change in the hormone levels of the body, where you have an increase of those tropic hormones. You're going to start to mature your gonads. So the sperm for males are starting, the spermatogenesis is going to start. And then for females, you'll start to recruit and mature the oocytes that are in the ovary. And then the woman will actually start her menstrual cycle, and then she'll be able to get pregnant. Other things happen at puberty, of course, secondary sex characteristics. So the hormones that are secreted at this time, the increase in hormone concentration will also bring the onset of, for females' breasts, the hips will get wider, you'll have pubic hair, things like that. And then males, again, pubic hair, increase the muscle mass, the larynx, your vocal cords will get bigger, so that deepens the voice. So he brings out all those secondary phenotypic traits of puberty. Now, in middle age, there's a little bit of a split where, in terms of gametes for women reach menopause. And the most common age bracket is somewhere from 45 to 50. You start this onset of menopause. Every woman's different. Some women actually have a very early menopause. Some women have late menopause. But this is a drastic decrease in estrogen, and this is going to have a drastic decrease in the ability to ovulate. So ovum are going to stop being matured, and so the woman can no longer get pregnant or have children. In men, this doesn't happen. Sperm is produced all throughout their life, so they are able to father children at any age. However, there is, right, if you see from this Time magazine, there's a lot of research out there that there is a shift. I mean, there is a shift. The testosterone levels do drop, but they think that there's also a lot of other physiological things that go into that. And they're calling it things like androgenopause or andropause or menopause. But you do have a dip in testosterone levels around the same time. Okay. But basically, it's just not as drastic for women. Women cannot have children anymore, but men are still making viable sperm. All right, so this is just a look at the reproductive tract. So I'm just going to go over this really quick as a review from anatomy, and then we'll go on to talk about the Sertoli cells and things like that. So here is our testis, right? So this is the gonad of the male reproductive system. This is where spermatogenesis happens. Right, we're making sperm, but also there are cells here that secrete hormone, right? So there are endocrine cells present. They're called the interstitial cells of Leydig. The reason why your scrotum, the reason why the testes are in a scrotum and the scrotum are outside the abdominal cavity is because the temperature of the abdomen is too warm for normal sperm development. So the sperm have to be at least 3 degrees or about 3 degrees cooler than the abdomen in order for the sperm to develop properly, which is why males evolved a scrotum to have the sperm outside the abdomen. As the sperm are made in the testis, they're going to enter into another structure called the epididymis. The epididymis is where the sperm will mature, and which basically means they gain the ability to become motile. And then they basically enter here at the head region. This is considered the head of the epididymis. The central part is called the body of the epididymis, and then this tail of the epididymis is at the distal end. When the sperm have gone through the epididymis, this takes about 2.5 months, according to Wikipedia. Then the sperm enter the vas deferens and stay at the lower end of the vas deferens until ejaculation. When ejaculation occurs, the sperm moves through peristalsis up the vas deferens. It moves into behind the bladder and comes to this junction point with these glands, these accessory glands that produce the semen. So the seminal vesicle and the prostate gland both produce substances that contribute to the semen. The sperm and the semen fluids are going to come together at the ejaculatory duct, which is this area here. And the ejaculatory duct then is going to be expelled through, again, skeletal muscle and smooth muscle contractions out of the body. We see a bulbourethral gland. This gland is the lubricant known as pre-ejaculate that is formed to both clean out the urethra and provide lubrication for intercourse. So this little gland here sits inside a muscle called the urogenital diaphragm. And that's pretty much it. So one thing to notice is that the prostate gland does encircle the urethra. So an enlarged prostate gland or prostate cancer tends to come with symptoms of urinary problems because it compresses the prostatic urethra that runs through the prostate gland. All right, so let's take a look at our seminal vesicle in the testes. So the testes are your male gonads. They are 80% seminiferous tubules. So the bulk of your testes are these tubes, right? They're called seminiferous tubules, where sperm is produced. And again, the name for sperm production is called spermatogenesis. And what we see here in this histology slide is that we have this circular structure. If we continue that picture, this is our seminiferous tubule. It's a cross section of it. We can see that we have different stages of sperm development, and we also have a cell called Sertoli cells, which I'll talk about in just a second. Outside of our seminiferous tubules, we have cells as well, which are called Leydig cells or interstitial cells, and they are going to secrete hormone. They are endocrine cells secreting testosterone. And then the Sertoli cells are present within the seminiferous tubule, and they're necessary for sperm development. So let's take a look at our Sertoli, let's quickly take a look at just a more detailed version of the testes, right? So this is our seminiferous tubules. You can see the bulk of the testes is seminiferous tubules. We have structures called the reedy testes and efferent ductules leading to the epididymis. And again, the epididymis is where these sperm are stored and then, matured and then stored in this last portion in the tail and also the first part of the vas deferens. So this is just a look at the cross-section of our seminiferous tubule again, noticing that in the lumen, this is where we have our sperm. So as sperm are developing, they are being pushed towards the lumen of our seminiferous tubule. So you can see the lumen, and that's where we find the sperm. We can see the Sertoli cells are quite large, and outside of our Sertoli cells, outside of our seminiferous tubule, we have our Leydig cells, which they color-coded in blue. Okay. Moving on to the next slide. I couldn't do that. All right. So let's talk about Sertoli cells. So the Sertoli cells are pink here. Okay. And they're going to do a lot of things. The first item in your outline is that they create a blood-testis barrier. So there are tight junctions between the Sertoli cells. Actually, they are pictured here too, right? This tight junction. And the reason why there are tight junctions present is to create a membrane that is going to restrict the fluid coming from the bloodstream. So the bloodstream is going to be out here, right? And the things in the blood, not everything wants to reach the developing sperm, because there might be some items in the bloodstream that will harm the development of the sperm. So this is an extra barrier that restricts movement of certain materials, because we don't want to have just anything bathe the developing sperm. So it's a controlled environment that the Sertoli cells set up with those tight junctions, that nice tight membrane. Number 2 is that they're found inside seminiferous tubules, and they also support sperm development. So they actually secrete substances that help the sperm develop. They also secrete the luminal fluid, so the fluid inside of the seminiferous tubules that the sperm are going to travel in as it's pushed through peristalsis. The fluid's going to move, and that fluid is created by the Sertoli cells. The Sertoli cells also make androgen binding protein. So here we go with androgen binding protein. So remember, your androgens, your estrogen, testosterone, there are steroid hormones, and steroid hormones are lipophilic. They like fat. Another way of thinking that is hydrophobic. So they don't like to dissolve in fluid. Remember, there's luminal fluid everywhere, which is mostly water. So the steroid hormones don't like to stay in the water. So these binding proteins allow the hormones to stay in the fluid longer and to give them more of a lifespan and maintain a steady concentration of them in the fluid. These are target cells for both testosterone and follicle stimulating hormone. So FSH is going to stimulate the Sertoli cells, as well as testosterone. And testosterone, which I talk about a little bit later, is necessary for proper sperm development, but so is estrogen. So these Sertoli cells will also convert some testosterone to estrogen. I talked about the blood-testis barrier, and then they're also lastly going to secrete inhibin. Inhibin is a hormone that's going to provide a negative feedback to the anterior pituitary so that we don't overload the testis with follicle stimulating hormone. The next cell here is our Leydig cells, our interstitial cells of Leydig, which are our endocrine glands, and they respond to luteinizing hormone. When luteinizing hormone is detected, they're going to increase their secretion of testosterone, and there's 2 forms of testosterone, at least here. There's testosterone that the sperm need to develop, and then there's a more powerful, robust form, I guess, of testosterone, which is DHT, which stands for dihydrotestosterone. Anyhow, it's a more powerful form of testosterone, and it really kicks off the secondary male characteristics. All right, so moving on to our accessory glands. I already talked a little bit about this, but again, the seminal glands or seminal vesicles are going to secrete our semen. And so let's just talk about the parts here. The alkaline fluid that is found in what the seminal vesicles create is to neutralize the acid of the vagina. Neutralize acid pH, right, of the vagina. Because the acidic pH can kill those sperm, and so we want to allow the sperm a fighting chance once they reach the female reproductive tract. Fructose is going to be energy source for those mitochondria, right? The mitochondria are going to use a lot of ATP, or they're going to produce ATP for the sperm to move, to swim, to move the flagella. And they need a source, a sugar source to burn, so that's the fructose. Prostaglandins is required for, it's thought to reduce the immune reaction on the female side, so that, because sperm is a foreign cell coming into the female's body, so the female immune system will fight off the sperm. So it's just a way to decrease that from happening, and also increases the motility of sperm. The prostate gland secretes citrate enzymes, and then I have down also zinc, and although unknown what zinc really does, if you have low zinc concentrations, men tend to have infertility. And then lastly, we looked at our bulbourethral glands, which is where we secrete pre-ejaculate. Okay, so I pointed those out in the other slide. Okay, so moving on to our hormone regulation. So here's just the flow chart of the topics we've talked about, where we have our hypothalamus ultimately producing our gonadotropin releasing hormone. The gonadotropin releasing hormone through the bloodstream goes to the anterior pituitary, and the anterior pituitary secretes two hormones, follicle stimulating hormone and luteinizing hormone. The follicle stimulating hormone is going to act on our Sertoli cells, and they're going to act, so the Sertoli cells are going to increase, they're going to promote spermatogenesis, so they're going to start to secrete substances that help those undergo meiosis 1 and meiosis 2. They also secrete inhibin, right? So inhibin, here's our negative feedback loop. So the inhibin is going to come act on the anterior pituitary so that we don't, so that we always have a constant level of follicle stimulating hormone. So we don't overproduce follicle stimulating hormone. It's a way to feed back and mediate the secretion. Luteinizing hormone comes in and acts on your Leydig cells. The Leydig cells produce testosterone, as they do, and the testosterone acts on the Sertoli cells, and as well as increases your plasma concentration of testosterone, because testosterone does have an effect all over the body, on muscles, on metabolism, and so testosterone levels are to go into the bloodstream. Testosterone also feeds back, right? So in the bloodstream, testosterone is going to feed back to both the anterior pituitary and the hypothalamus, so that we're not going, so we keep a constant level of testosterone, not too much. So these are just some to round out the view of androgens, namely testosterone in males, to stimulate spermatogenesis, to promote development of our secondary sex characteristics at puberty, to increase the sex drive, to promote protein synthesis and skeletal muscle, right? So this is why people like to use steroids. When they say we're using steroids, they really mean androgen testosterone, because it helps build muscle and makes people stronger. Stimulate growth hormone secretion. Again, the building anabolic function of testosterone is synergistic with growth hormone, and it promotes development of the male reproductive structures in the embryonic life. We talked about that at the very beginning of this, how testosterone helps form those Wolffian tracts. All right, so taking a look at spermatogenesis. Okay, so let me just make sure I've covered things. Okay, just in terms of the blood levels of sex hormones in males, what I'm trying to say here is that the blood level hormones in males, even though it pulses every 2 hours, it has 2 hour cycles, throughout the day, it's pretty even in the body, which is not the same for females, because females will have a monthly cycle. So the levels of estrogen versus progesterone versus luteinizing hormone versus, like, they all shift over the course of her monthly cycle. But for men, it's pretty constant. And then prepubescent boys will have lower levels of testosterone, and then that comes up at puberty and adulthood. They have higher levels of testosterone, and and levels of testosterone also varied. So some men have lower levels of testosterone than others. Some men have higher levels of testosterone than others. So that level is actually individual for people. All right, let's take a look at sperm development. So the process of developing sperm, again, is spermatogenesis, and this happens inside the seminiferous tubules of the testes. Here we are at our spermatogonia. So this is our stem cells. Our stem cells are 2N, right? They're diploid. And you're born with all the spermatogonia that you'll need, and then as they are recruited to undergo meiosis, you don't lose them, because they do replace themselves with mitosis. So there's a replacement. And this is why men can father children all their lives, because you constantly have this renewable supply of stem cells. So as the spermatogonium start to get recruited into the meiosis pathway, the first maturation process is forming a primary spermatocyte. So primary spermatocytes are actually still 2N, So we haven't really done anything with the chromosome number yet. But the primary spermatocytes enter the primary phase of meiosis, which is meiosis 1. After that, you get a secondary spermatocyte. So actually you get 2, right? So you have 2 secondary spermatocytes. And those spermatocytes are now haploid. So after that first round of meiosis, you're already haploid. Then those secondary spermatocytes undergo the secondary phase of meiosis, meiosis 2, and they're going to form, because they're splitting again, they form 4 spermatids. So spermatids are also N, they're also haploid. At this point, we're done in terms of the chromosomal change. Meiosis is finished, but we see that the morphology, the shape of these cells is not like a sperm. They're still round, so we have to change the shape of our spermatids into what we know as spermatozoa or sperm, and that process is known as spermiogenesis. Okay, so step 4 would be spermiogenesis, where you have that shape morphological change. And then we see again how it's pictured, where we start at the periphery, the stem cells are at the periphery of our seminiferous tubule and move towards the lumen, and we find the sperm at the lumen of our seminiferous tubules. So after spermiogenesis, you have what we see here, a sperm cell, and the sperm cell has a head, a midpiece, and a tail. The head has a nucleus, and that's where our chromosome lie, and then the acrosome is a special sac, membrane-bound sac, that contains hydrolytic enzymes that will help to digest their way into the egg. What's interesting is the acrosome is destabilized. So this acrosome, once the sperm gets into the female reproductive tract, there is a biochemical change in response to substances in the female reproductive tract, and it's called capitation. I couldn't say that word. No, capacitation. Anyhow, we're destabilizing. I can't talk today. Destabilize the membrane of our acrosome, and the acrosome will be more easily broken open. And so when that happens, the enzymes come out more easily and can eat their way into the egg. The midpiece has mitochondria, as we can see, so this is going to power our flagellum, so it's going to make ATP, and this is where the fructose comes in, the fructose supplied by the seminal vesicle. It's going to allow the mitochondria to undergo oxidative phosphorylation and make ATP, which then allows the tail to move and swim towards the egg. All right, so in terms of where sperm maturation occurs, so we have development in the seminiferous tubules here, right? And so that's going to take about 20 days until they move into the epididymis. So through paracelsus, smooth muscle contraction, and the flow of our fluid produced by our Sertoli cells, they're going to spend about 2.5 months in the epididymis. And that's where they mature into gaining the ability to move. So their flagellum are going to be fully functional. And then they're going to move to the vas deferens, which is our last area here, right? And also by paracelsus, and remain there until ejaculation. Okay, here's that word, capacitation. All right. So the sexual response in males is our last topic. This is controlled by the autonomic nervous system. So remember, the autonomic nervous system is part of the nervous system that it's not voluntary, it's completely involuntary. Special response is actually a spinal reflex. And the phases include erection, emission, and ejaculation. Erection is controlled by the parasympathetic nervous system. And this is your rest and digest part of your nervous system, your autonomic nervous system. And so you have to be relatively relaxed in order to achieve an erection, or you have to at least be in that parasympathetic state. So then we have emission and ejaculation, which are both sympathetic. So there's a shift from parasympathetic to sympathetic. And the stimuli for the sexual response is mechanoreceptors, so touch receptors of the penis, and then other erogenous zones. And then also just descending signals from the higher brain centers. So just, the psychology, what you're thinking about, that's going to also be a stimulus. So if we take a look at this, mechanical stimulation, descending neural pathways from the brain, coming into the spinal cord. So the spinal cord is going to control your activation of the parasympathetic nerves. If you downregulate the sympathetic neurons and upregulate, I shouldn't say downregulate, upregulate. Just basically reduce the activity of the sympathetic neurons and increase the activity of parasympathetic neurons, then you're going to allow the blood vessels to dilate. So this is the key. The arterioles supplying blood to the erectile tissue is going to have a relaxation of smooth muscles. This is going to dilate those blood vessels in the penis. The penis has erectile tissue, which has vascular spaces. And as the blood moves into the penis, those vascular spaces will fill with that blood and cause that tissue to expand, causing an erection. So erectile tissue has an increase in blood flow. What happens here is that you have an increase of blood volume and blood pressure in the penis. So you would think that blood coming in is brought by the arteries, right? In this case, the smaller arteries called arterioles. But you also have another network of blood vessels that bring blood away, which are veins. But what happens is, as you increase the pressure inside the penis, the veins start to collapse because the walls of veins are relatively thin. So a little bit of pressure will start to collapse those veins. So you have a compression of the veins, and that's going to restrict the amount of blood leaving the penis. So you're going to reduce the outflow of blood. This is a positive feedback loop, because if you're decreasing the flow of blood out and you're continually having blood come in, right? So what happens is you're going to increase the blood volume even more. You're going to increase the blood pressure even more in the penis. With that increase in more pressure, you're going to have more compressed veins, decreasing the amount of blood that can leave. And then you can see how this is a positive feedback loop. So that's where how an erection is maintained. Even though you have blood coming into the penis and leaving the penis, the blood leaving the penis is very, very little. And here we can see that ultimately this leads to erection. All right, so in terms of sexual response for emission means that we have the contractions of our tract, so the epididymis, the vas deferens, and the ejaculatory duct. They're going to move the sperm and also the semen into the ejaculatory duct. So secretions from the seminal vesicles and prostate occur. So remember, the smooth muscle in those two glands will contract, secreting their semen. The result of emission is that we're moving the semen, which is sperm and the fluid, into the urethra. So we're basically getting ready for ejaculation. So again, this is sympathetic. Nervous system needs to do that. And then ejaculation is again sympathetic activity. So again, a response to mechanical stimulation continues to until ejaculation. And here, when we have ejaculation, we have contraction of smooth muscles in the urethra, but also skeletal muscle at the base of the penis helps. And at the same time, remember, this is a reflex. So there's multiple things going on out of your control. It stimulates the closure of the urethral sphincter so that there's no accidental leak of urine into the sperm, because urine can harm the sperm. All right, so the result is that we have movement of semen through the urethra and out of the body. All right, so that's it for the male reproductive system. Let's have some wrap-up questions to think about, or do some wrap-up questions. Remember, you can pause it to think about these yourself. Which of the following components of the male reproductive system is directly responsible for the synthesis and secretion of testosterones and other androgens? So pause. It is Leydig cells. Which of the following is not a function of Sertoli cells? Go ahead and pause it. The answer is B, right? Sertoli cells don't make and secrete luteinizing hormone. That is the job of the anterior pituitary. Which of the following is not a function of testosterone? The answer is C, stimulating the secretion of gonadotropins, because they don't. Testosterone actually feeds back negative, and it would actually inhibit. the secretion of gonadotropins. Which component of the mature sperm contains enzymes and other proteins essential for fusion with the egg? And that's going to be acrosome. And that's it.